Today we're going to be talking about blasting a gene, which basically just means that we're entering a protein sequence into a database, which is going to then compare established genes with their respective functions into the system and compare it to our protein sequence. When we blast a protein sequence, the database is looking for sequence homology. This means that they're comparing the protein sequence of our phage to the ones on the site and seeing if they're similar at the amino acid level. This can be used as evidence when assigning a certain function. It's important to note that sequences can be homologous or similar at the protein level while differing at the DNA level. This is because several codons may code for the same amino acid. The first thing that you're going to do to blast a protein sequence is pull up the Phagedb site, which you can see here. For our purposes, we are going to be searching the phage SOB, but y'all are going to use the phage whose genome y'all are annotating, which is going to be Iceman. So after you search the phage SOB, or Iceman in y'all's case, it's going to pull up a page that looks similar to this. There's a lot of good data found on this page, but we are going to be scrolling to this tab here that says Gene List. From here, you can choose the specific gene that you're working with. So we're going to be looking at gene 14 as an example. Just remember that draft means that no one has annotated this genome manually yet, and so you're going to click here. This page has a lot of useful information. Here you can see how long the gene is, and here's the entire amino acid sequence. These two tabs at the top blast the protein sequence. To blast through Phagedb, click this button here. Phagedb is a more localized database compared to NCBI, which you can see here, but both are important for supporting a certain function in a phage, and we need both to be utilized as evidence for that function. We're going to start with Phagedb. So here's that amino acid sequence again, so we are going to click here to blast. This is what the blast page of results should look like. These red lines basically just help you visualize what the alignment is looking like. It helps you gain a little more understanding towards what the blast has done. This comparison is clearly very similar, so all of the red lines look the same size, but in other blasts they might not. When you scroll down, you'll see all the phages that the database has found sequence homology with. So since draft means no one's looked at this genome yet and taken each gene and annotated it, we aren't going to use drafts as evidence for a function because they're not as reliable as the ones that have been annotated manually. So the first hit is our phage, which would make sense because it's going to align completely with itself. The next one is a draft, which isn't as reliable, and the next one is a phage called Donkey Kong that has been called to have a tape measure protein at this gene. So just by scrolling down, we can see that when a phage isn't labeled as a draft, that is going to be the function called, and so clearly I would feel pretty good about calling it as a tape measure protein since there's substantial evidence. So now we're back on the original Phagedb site that we just used, and we're back on that gene details page that is discussing gene 14. So again, this tab here is how we got to that Phagedb blast site. The NCBI tab is right underneath, and it does a more widespread blast to compare our protein sequence with a wider range of protein sequences in a database. So we're going to click here next. So again, here's that protein sequence. It's important to make sure that you're on the blast P option at the top rather than any of these other tabs because this website has a lot of different purposes. So we're just going to scroll down here and hit blast. This is what the results page should look like. There's clearly a lot of good evidence for the tape measure protein being called, so I would feel good about making that call. You can see here the e-value is noted, and e-value is just a way to measure whether or not the data found is reliable or not. The closer the e-value is to 0, the better. It's also important to note that where it says percent coverage or percent identity, that's just another term for homologous coverage, so it's measuring the similarity between our phage and the hit. So you're looking for a high percentage point of coverage, preferably as close to 100 as possible. So since these are all in the 90s, that would be a reliable call. So that's how you complete a blast, which again is just taking the amino acid sequence and comparing it to a protein sequence of a previously annotated phage. If a phage is being blasted, it's shown to be genetically similar to one that has already been assigned a function, and we can use that phage as evidence to show that they may share a function. This allows us to have more information about our phage's genome.